if you're calling in.
Yep, I'm doing it. I've got it now.
Cool. Appreciate you. All right, so yeah, let's go ahead and get started. So hello, everyone, and welcome to another exciting installment to the Computational Toxicology Communities of Practice. My name is Sammy Hamp. I'm a public affairs specialist here in the Center for Computational Toxicology and Exposure. Today we'll be hearing from Jennifer Olker and Colleen Elonen with CCTE about modernizing the ECOTOXicology Knowledgebase. Before we get started, I just want to ask everyone to please mute their lines. You can accomplish that by hitting #6. And then without any further ado, here's our presentation.
All right, thanks, Sammy. This is Jennifer Olker. I'm a postdoctoral researcher at the Great Lakes Toxicology and Ecology Division in Duluth, part of CCTE. And my co-author on this presentation, Colleen Elonen, is on the line, but I'll be doing the entire presentation as we weren't sure she was going to be able to join us. So since I'm doing the presentation, she gets all the hard questions. Thank you, Colleen, for calling in. So I'd like to thank you all for joining us today, and on behalf of myself, Colleen, and our entire ECOTOX team, I'm excited to share with you some of the updates of where we're going with ECOTOX and some of the efficiencies that we're working towards to get through more literature reviews faster and get data out to folks even faster than we do now. I'm assuming everyone can hear me unless someone tells me otherwise. So first, I'm going to provide a little bit of background and history for the ECOTOX Knowledgebase, but spend most of the time today talking about our recent efforts to modernize and improve efficiency of the ECOTOX pipeline from chemical identification and literature searches through review and data curation, and then to talk about our recent efforts to identify ecological toxicity of per and polyfluorinated substances with the ECOTOX protocols. These topics were scheduled to be presented at SOT by Colleen and myself, and so it's great to have a venue to share that information. So if you don't already know, ECOTOX is a comprehensive knowledge-based system that summarizes toxicological data from single chemical exposure studies to terrestrial and aquatic organisms. We cast a broad net to capture all pertinent literature from sources such as journal articles, gray literature like government documents, and theses and dissertations. We are continuously reviewing articles for inclusion, and the knowledge base is updated quarterly to the public website. So you can see here our last update occurred on March 12th, 2020. And so with that update on March 12th, we now include over 12,000 chemicals for over 13,000 species and over 50,000 references, and are getting close to 1 million test records in the database. We have an over 30-year history of curating data and reviewing the literature, and are supported by numerous EPA program offices and the Office of Research and Development. This user interface you see here is based on the major update that went out about 1.5 years ago. So we completed some big improvements there, and we are continuing to work on new functionality and improved interoperability with the database. So data from ECOTOX are used from a variety of applications by multiple types of users with thousands of distinct IP addresses accessing the database each month. We don't require logins or documentation of uses, but we can get a sense of the distribution of users based on those that contact the ECOTOX support line. And this figure is a little outdated, but it still reflects the distribution that we see today, with clients including the private sector, actually making up a big portion, state government, local governments, other federal agencies, US EPA and EPA contractors, as well as academia. Just somewhere on here. Yeah, here we are. Within the EPA, data from the ECOTOX knowledge base is used directly in environmental decision-making by program offices and regions. For example, ECOTOX is the source of data for all ambient water quality criteria since 1985, and the Office of Pesticides and Ecological Fate and Effects Division uses ECOTOX as the source for data for all ecological assessments. And data within ECOTOX are also used for site-specific ecological risk assessments and are routinely used for Regional Resource and Conservation Recovery Act programs. Since early 2017, we have been working with the Office of Pollution Prevention and Toxic Substances to be the source of environmental toxicity data to meet the objectives of the Lautenberg Chemical Safety for the 21st Century Act. And over the last 2 years, we've conducted a major effort to provide ecological toxicity data for PFAS to researchers, the Joint Work Group of EPA Ecological Risk Assessment Forum, and the Department of Defense Environmental Risk Assessment Work Group, as well as others. And that last piece I'll be talking about more at the end of the presentation. So as I just described, ECOTOX data are used by federal, state, tribal, and international agencies for a variety of applications as listed here. But we also know that data from ECOTOX are a component of many tools and applications, including tools for species sensitivity distributions, predicted no-effect concentrations, quantitative structure-activity relationships, bioconcentration factor modeling and validation, and development of adverse outcome pathways. Currently, one of our main focuses is working towards increased interoperability with other databases and tools through incorporating relevant unique identifiers to make these links, as well as investigating options to provide web services for ECOTOX. So I'm going to show you kind of an overview of how the data are identified and curated, and to provide context for the folks familiar with systematic review, here I've provided the general PRISMA steps in gray, and then the ECOTOX pipeline steps in blue. So we start off with chemical verification to look for the chemical name and CAS number, but not just a single name. We also look for synonyms and trade names and other relevant forms as requested by the client for which we're doing a literature search. We use a variety of sources for that. Literature searches are then conducted through a variety of different search engines, including ProQuest, ScienceDirect, Agricola, Web of Science, as well as our internal EPA database where we've been collecting references for many, many years. These numbers in here are just an example. So the citations are downloaded from these sources, we remove duplicates, and then conduct title and abstract screening. At the title and abstract screening, we're identifying things that are applicable for ecological species and a single chemical exposure. So those that don't meet that applicability criteria are excluded and are tagged for the reason of exclusion for reviewing again in the future, or to provide to other researchers who might be interested in chem methods or in human health or in fate data. And then the references move on for full text screening, at which point they are reviewed in more detail and looked for quality control metrics and acceptability, such as including a control, having a biological endpoint that's being measured. And finally, for those that meet the applicability and acceptability criteria, data are extracted and included into the database. So I'm going to provide a little bit of, oh, I forgot about this. While we don't have a distinct study quality evaluation step in our pipeline, many of the fields that we extract and include in the database are relevant for answering some of the standard study quality evaluation questions, and I'll talk about that a little bit more in a few slides. So to provide a little bit more detail and to highlight some of the improvements that we're working on, I'll talk about each step on a slide. So we're looking for these, they're developing the chemical search terms. So we go through a chemical verification process using a variety of sources, finding synonyms, verifying the CAS numbers, and eliminating poor search terms. This historically has been done manually to develop a list of search terms as seen here, and takes a few hours. However, we're currently working with the EPA's Environmental Modeling and Visualization Laboratory to develop methods for computer-assisted identification of these search terms to better document where the search terms came from and really improve the speed and transparency of this process. So this is in place now. These search terms are then used through multiple search engines, as I mentioned, collecting the data, removing duplicates, and then going for review. In 2019, 159,000 plus references were manually skimmed for applicability at that title and abstract level. To improve this process, we are working with SciOM to develop machine learning and natural language. Sorry, I missed a step here. To improve this process, not the next one, we are working with EMVL and the developers of Abstract Sifter to modify and expand on this Excel-based program to expand it beyond PubMed, and then also to allow options for batch searching and exporting the references as RIS files so they can be easily imported into other programs. So this is where I had skipped ahead to a second ago. So at the title and abstract level, we're skimming for applicability. Again, historically, this has been done manually with human eyes on the title and abstract. However, we're working with SciOM to develop machine learning and natural language processing for the literature review at this title and abstract skimming level, modifying the existing Swift Active screener with ECOTOX specific strategies and criteria, such as including the reason for excluding a paper. Then the full text is reviewed for those that pass the title and abstract screening. And this is still a manual process, but it includes human eyes looking at things. However, we are exploring options for data mining that will pull out relevant terms, highlight those of interest, and also some options for computer-assisted review, and in the future, computer-assisted data curation. So just to give you the list of things that meet the applicability criteria, to be applicable for ECOTOX, papers must be for a single chemical exposure to an ecologically relevant species. We have to be able to verify the CAS number of the chemical exposure and verify the test species. This has historically been to live organisms. However, we are expanding this as there are increasingly viable tissues or cells of ecologically relevant species that are used in vitro studies. There also needs to be a reported exposure concentration, dose, or application rate, and a reported duration of exposure. And very importantly, there must be a control in the study. If there's not a control, it does not meet the acceptability criteria to be included in ECOTOX. So some examples of things that aren't included are review studies, studies on humans, monkeys, bacteria, viruses, and yeast, air pollution studies, and we have a challenge in including non-English publications, although in some instances there is a good reason to deal with translation efforts because there are some studies that are very relevant. So for those that are excluded from inclusion in ECOTOX that we've looked at, we tag them with the reason that they were excluded, whether it be that there's only an abstract or that the CAS number was unavailable, it was a non-English paper, or perhaps it was already published as somewhere else. And there's a variety of all these tags, so we can always go back and identify why a paper was excluded from inclusion into the ECOTOX knowledge base, and also identify those papers that are relevant for other research areas. So then we get to the data extraction step. For acceptable papers, up to 250 different fields of information are extracted from each paper as relevant for that reference and is reported by the authors. This includes unique identifiers for the chemical and the species, as well as some other information for the test species, as well as the chemical, such as concentrations, chemical formulation, was it a nominal or verified concentration, and then also a wide variety of information and details about the study design and the test conditions, as well as everything about the results. So as you can imagine, this is quite a feat and is done manually through a really nice system that was developed by the programmers that work on ECOTOX. However, we are exploring ways to do some computer-assisted data extraction and data review with some automated data extraction options out there. We have not made very much progress on that. And along those same lines, we are incorporating some of these standardized unique identifiers. We've already done this for chemicals and species, but there are also standardized unique identifiers that we can be adding for genes and for proteins. So we're working towards that to increase interoperability for the data that has already been extracted. Additionally, we are just in the beginning stages of developing ECOTOX ontology, and the ultimate goal of linking the effects in ECOTOX to biological pathways, as well as all of the other wealth of data that are available. So then we get to this question of study quality evaluation. It's not a discrete step in the ECOTOX pipeline, but many fields in ECOTOX can inform study quality evaluation questions that I have found. Some of them actually are required for inclusion in ECOTOX. So this includes, is the test substance identified? Well, yes, that is a requirement for inclusion in ECOTOX. Is the species given? Yes, again, that's also a requirement for inclusion in ECOTOX. And a control is required for inclusion in ECOTOX. Many of the other questions that I've found in study quality evaluation guidelines, ECOTOX has fields that can inform those, such as the test methods, the organism source, what type of chemical analysis was done, what was the statistical significance and the significance level conducted. We also recognize that there is a need to have a unified method for study quality evaluations within the agency, and are working with both OPPT-RAD and the Office of Water on pilots for compiling study quality information. So this is done for the pilot with the first 10 priority TSCA chemicals, where the ECOTOX team worked on identifying and evaluating studies based on the critical domains of test substance, test design, exposure characteristics, test organism, outcome assessment, confounding and variable control, and the data presentation and analysis. And we're working on some initial DER templates with the Office of Water that have a lot of the same critical domains for evaluation. So this is just a first step, but we're definitely moving in the direction of incorporating the more necessary fields for evaluating study quality. Just saw a little message pop up. Is everything displaying and the sound okay?
Yep, we're all good.
Okay, thank you. So that concludes the background and the modernizing efforts that we have underway. We're happy to take questions for those at the end, and very happy that Colleen is on the line so she can address some of the detailed questions. So now I'm going to move on and share an example of how we have applied the ECOTOX pipeline in the last few years for some priority chemicals, specifically for the per and polyfluorinated substances. So as I'm sure everyone on the line knows, perfluorinated compounds are of concern, including possible water contamination and potential exposure of aquatic and terrestrial life. And there are drinking water health advisories for PFOS and PFOA relevant to human health, but there are not yet ecological receptor-based benchmarks or criteria, despite the fact that there are evidence of exposure, there's a potential to bioaccumulate, and there are a variety of other effects documented on ecological species. Thus, there is a need for ecological toxicity data to inform risk management, risk assessment, and management, and not just for PFOS and PFOA, but really for a broad range of PFAS. And so identifying what information is out there is something that we can apply the ECOTOX protocols to, and we have a system and a well-defined process that is both transparent and comprehensive. Therefore, our objectives were to identify and describe the available empirical evidence for ecological effects of PFAS, and then next, to start using this information to identify potential ecological toxicity pathways for these compounds. So we have been working on this for about 2 years officially, and as PFAS data have been identified through the literature, they've been added to this public-facing website. Pretty much every quarter, PFAS have been included in this top list of recent chemicals with first full searches and coding completed. So you can see that they are right here, and if you hover over there, you can actually click. These are all hyperlinks. You can click right out to explore the data that are available. So we'll do that just as a quick demo. And we can see that currently, so the most recent update, we have data in ECOTOX for 112 PFAS. Can't see them all here, but there's a long list. Some of them have a lot of information and some of them have just a little bit. We click on this plot view, we can see that for those 112 unique CAS numbers that are PFAS, there are 18,140 records. So this will be a little bit of a challenge to sort through if you just wanted to skim it all, browse it in an Excel file. But if we think about what type of information are there, we can use the plot feature in the Current Explorer to get a sense of what information are available. Here we have on the y-axis the concentration of exposure in a standardized parts per million, and on the x-axis we have the general effect type that was measured in the studies, from biochemistry, enzyme, hormones, you keep going through to growth, morphology, behavior, reproduction, mortality, and population. So there's a wide variety of different types of effects that have been measured across a range of concentrations for these 112 PFAS compounds in the literature. So that is a little much to see on the web browser and way too much to manage in an Excel spreadsheet to look at. So I'm just going to provide some simple summaries, but first want to go back to the literature search and review flow diagram to understand how these data were identified and reviewed. Where did they come from? So we started off with the chemical-based search terms, and this is a ever-changing list. It currently is just over 300 chemical names and CAS numbers, as well as a set of generic PFAS terms. And I'll talk a little bit more about those in a minute. And then we conducted those literature searches, and this is the accumulation of multiple searches that have been done over the last 2 years, as we have been updating this information quarterly. So through these literature searches over the last, as I just said, 2 years, we have downloaded 145,000 citations approximately, removed duplicates, which led to 16,091 references in title and abstract screening. Through that process, many of them were found to be not applicable and thus were excluded because they were chemical methods relevant only for human health. We have a large set that were survey data and have been forwarded to folks looking at that information, as well as some other duplicates that weren't identified through the initial duplicate removal process. 565 references moved forward for full text screening. Some of those don't meet the acceptability criteria, and we ended up through this process with data extraction from 462 references and a total of 13,496 records. However, that doesn't match the total I just told you because we actually already had some relevant PFAS references in ECOTOX, and not just some. We had about 192 before I started documenting the focused PFAS effort here. So currently there are 654 references for PFAS in ECOTOX with data extracted. And these 654 publications are across the 112 PFAS. And here I'm just showing a distribution of where the information is based on the number of references, with the box size representing the number of references for each compound, and they're separated out by CAS number. So this is potassium PFOS, this is PFOA. But then you can see if this green box here is potassium PFOS, this is the PFOS ion, and this is another PFOS CAS number. So these three boxes are really practically the same compound. You can see most of our information is still really just for PFOA and PFOS, with a few other compounds growing in the number of publications that we have in ECOTOX. But the vast majority of the compounds have only one or a handful of references. So it's still very limited information available for ecological toxicity data. And how does this fall out for these 112 across the family tree of PFAS as described by the OECD or in Wang et al. 2017? We know that there are multiple subclasses of PFASs, and how do the 322 chemical names and their associated CAS numbers fall out into that? Well, we attempted to search based on interest of the EPA research group that had started off with a list of PFAS. And then we've added to that list ongoing, but really most of the search terms that are included are for these subclasses that are in bold, where the subclasses that aren't in bold have either no or very few chemicals included in them that were part of our search. So we're encompassing more than just one subclass, but definitely not able to be entirely comprehensive at this time. And as you could see on the figure I showed with the PFAS and PFOA encompassing most of the references, you can see that even within some of these groups, like this top one, for some examples for PFOA, we have 128 references, but for some of the other compounds in that same group, there are very few references. And the same is true for the sulfonic acids, where potassium PFOS has the most records, and then something like PFHXS has only 5 references at this time. So we've been working on this for a while, as I mentioned, about 2 years, but I took this chart out earlier than that because Colleen Elonen, the ECOTOX coordinator, had insight before we were tasked with looking for PFAS that maybe it would be something that we should work on. So back before March 2017, there were about 100 PFAS references in ECOTOX. And then in, I think it was in October of 2017, Colleen used an internal ECOTOX list of PFAS to update what we had there. And then in April of 2018, we took an internal list from ECOTOX as well as a draft cross-agency list of PFAS of interest, which ended up with being about a list of about 200 perfluorinated compounds and their related CAS numbers, and that was conducted. So these are the time points of each of the searches. There's about a 6-month delay before the results from those searches end up in the data extracted and available on the public-facing website. So as you can see, you have a search done in April, but you don't see a major uptick in the number of references until maybe September or December of that same year. So another search was done in November of 2018 to include the ToxCast PFAS Set 1. There's only about 50 that we added there because the other 25 were already in our list for that list of 75 in Set 1. And then in July, August of 2019, we did a comprehensive list of everything that we had previously searched, but we also added any new chemicals that were in the more finalized EPA research list, as well as the ToxCast Set 2 75. And then when that ToxCast Set 2 and Set 1 got revised due to some stability and QA issues, we actually did a search in November of 2019 with just the 23 PFAS replacement chemicals that weren't already on our list. As you can see, this gray number of the total number of references is growing and growing. We expect it will continue to grow because where we're at right now here in the March update incorporates everything to this date with we always have about 30 papers that we haven't gotten to yet, but we also have done 2 additional searches in the meantime for a set of 24 priority PFAS that were based on the chemicals that are in the Superfund list, and that also encompasses the Department of Defense Tri-Services priority list. So this is going to be an ongoing process, but I just wanted to give you a sense of how frequently we have done the searches and how this process will continue. So then to think about that's the number of references, the number of chemicals, what does that mean is for coverage across biological species? As you can see here, this is a cumulative percent of records by the number of species. We have a diversity of different species types represented in this data set. It includes data for mammals, fish, amphibians, birds, mollusks, crustaceans, and a variety of plants also. However, about 20% of the records are for zebrafish right here. And then the next most frequent species represented is the Atlantic salmon, followed by Daphnia magna, followed by Chironomus riparius. So a lot of different species, but only a few represent almost 30% of the records in ECOTOX. However, within each of these groups, there are a diversity of different types of effects that are represented in this data set. Here we have the number of records by each species group, and then the general type of effects. So in the blue, we have about a third of the data are actually for fish, and of those, about a quarter of the records for fish are actually genetic data, typically changes in gene expression. But there's also biochemical data, mortality, morphology, growth, enzymes, hormones, behavior, etc. So fish are definitely the most represented species group, but there's also quite a bit of information for insects and spiders, as well as crustaceans, birds, and worms. These other groups get much smaller. So for the example for fish, we can look at the different types of effects, and we've got cellular responses really happening within the effect groups for genetics, biochemistry, enzymes, hormones, and cells. Quite a few records there. You get down to the other end where you get look at population. There's not nearly as many records on population effects. Organismal responses, including mortality, reproduction, definitely a wide variety of different effects measured. And each of these general effect groups can be broken down into more specific. So if we look just at this reproduction effect group, reproduction has a variety of different effect measurements. It could be measuring fecundity or fertility or hatch or number of spawns per female, and the list goes on. So we are going to continue to do this literature search on a quarterly basis with review and data extraction. So we anticipate having multiple references for PFAS incorporated into the public-facing website every quarter for at least the next 2 years. However, this is really, what I've described so far is really a data inventory. And what we want to try to move towards is some sort of summary or synthesis. And this is outside of the ECOTOX protocols, but will incorporate some of the aspects of interoperability that we're adding to ECOTOX at this time. So if there are 440 biological species and 112 different compounds in this group for over 1,000 different types of effects, how do we start to summarize and synthesize this information? If we just focus on one species, zebrafish, that has a lot of data, and one compound, this is PFAS, but think about all the measurements that might be available. We've started to try to come up with a strategy. And the first step is to start organizing the data in the framework for adverse outcome pathways, where you think about a toxicant might have cellular responses, tissue and organ responses, and organismal responses. And what we're trying to do is link some of these upstream cellular and tissue and organ responses down to the organismal responses. So if we think about increase in mortality, changes in growth and in behavior, can we link that back to some of the upstream effects that we're seeing by combining all of the data that are available? So in this example, I just pulled out some of the responses that we're seeing and show the structure that we're organizing the data. And so far, we've been doing this primarily as a manual process and review, with thoughts towards moving this to a more automated process. But there's still a lot of expert review that would need to happen at this level, given the current structure of the database. So we've started working with Rongling Wang, who's done some really interesting work recently to incorporate ontology-based semantic analysis into this process. I am not an expert in this area, and so I will defer any questions to Rongling. But the goal here is to be able to comprehensively identify potential toxicity pathways. And this process can help bridge the gap between the molecular and non-molecular phenotypes and lead to better understanding of the underlying modes of actions, while also allowing comparisons across chemicals and within and across species. So very briefly, if we have the data that's extracted from a paper and put into ECOTOX, and this would be in just some descriptions, it can then be used in ontology-based semantic analysis as demonstrated in this recent publication by Rongling, where you can annotate it and convert it to ontology classes, where for each species and chemical combination, you have a phenotypic profile. And those can then be assembled together and compared to existing phenotypic profiles that are out there, including those for genes such as that are available in ZFIN, pathways such as KEG and reactome, and disease pathways, or disease phenotypic profiles. However, this process is going to take some time, and we are moving in this direction and making some progress, but it is not going to be an immediate information, and there is definitely some interest in getting some immediate guidance on toxicity pathways that might be perturbed by this compound. So we decided to take an initial step by focusing on the biochemical and genetic measurements. Remember that in the fish data, about 25% of the effects were genetic, and so if we put the biochemical and genetic effects together, that represents about 40% of the total effect measurements in this data set. So just thinking about the zebrafish PFAS references, and this goes back actually to the update before our current one, because it's when I started this work, about 250 genes have been measured in zebrafish for changes in gene expression, and there have been 49 biochemical measurements taken. And so these are much easier to link to ontologies than the general terms and phrases that are used for effects at the organ or whole organism level, because we can take these and map them to standardized identifiers. For example, we can take the genes and map them to the Entree gene ID in NCBI, and then take those gene IDs and identify pathways through KEG or another option. And so as an example, we did this, and we found that there were 73 zebrafish pathways that were investigated based just on the genes that were measured for changes in expression. So this is what was investigated. However, only some of these actually saw significant changes in transcription. So this approach is what we are using to start to identify potential toxicity pathways. We still have a long way to go, and we definitely are going to be moving to a more comprehensive ontology-based semantic analysis, but this is an initial step. So in summary, we have really done quite a lot to expand the extent of literature on PFAS that are incorporated into ECOTOX, and summarized here is kind of the extent and distribution of what's available. We feel that this curated data is relevant for multiple applications, and one of the most interesting things is how easy it is to identify the data gaps when you go through this process. Definitely some areas that need more research, and others that really need to be summarized and synthesized. We've also identified literature that are available for other areas of PFAS research, such as BCF and BAF. And we feel that the ontology-based semantic analysis really could help us advance the synthesis and interpretation, not only for this set of priority compounds, but across the entire ECOTOX knowledge base, all the compounds that are in there. However, we also know that there are some limitations to what's included here, and some of the limitations are that we only include single chemical exposure. So mixtures are not currently included and extracted in the data for ECOTOX. However, papers that do have only mixtures are labeled as such and tagged, so we could go back to those. Also, observational and most field data are not represented here. And if we're just focusing on the genes and pathways, that's very limited for many ecological species, and so we'll be having to deal with that issue moving forward. So that is all I have for today, and we'd be happy to take some questions. And I say we because Colleen Elonen, the ECOTOX coordinator, is also on the line. You can also feel free to send myself an email. I've got my email listed there. And if you have questions specifically about ECOTOX data and access, you can call the ECOTOX support line listed here, or you can send them an email.
Okay, yeah, if you have any questions, feel free to put those in the chat and we will respond to you.
It looks like there's one from Ruth Ann. Do you want to read that,
Yeah.
Sammy?
Yeah, here we go. So we've got the first question. So in the first plot of PFAS chemical data, it looked like developmental endpoints had a very wide range of concentration effect levels compared with other endpoints. Did I read that correctly?
Yes, perhaps. I assume, I'm going to go back to that slide. I think that is in reference to this plot here. And I just want to point out something that I glossed over in showing you this. There's a total of over 18,000 records, but this little point here says that only 3,000 records will be shown on the plot, and we've organized it so that the first 3,000 that are shown are the lowest concentrations. So yes, we're looking at a log scale here for the y-axis, and so you can see that development and growth definitely had measurements taken at lower concentrations than these other effect groups. Definitely reading that right, but you have to be a little cautious because we're not actually able to see all of the data on this plot at the same time. It's just the lowest 300. And incorporated in these points, these could be low X, they could be no X, so that we're including no effect data as well as effect data. And that is an option that on this left-hand side query, you could go down and filter out so you would only see effects and exclude the no-X if you'd like, or just focus on LC50s. There are a variety of different options there.
Okay, and it looks like we have another question from Ruthin, and they want to know if you were to pick a small number of eco-test or endpoints to do for many PFAS to facilitate risk assessment across many of these chemicals, which endpoints should be prioritized?
So I think that is a really good question, and I am not the appropriate person to identify which endpoints should be prioritized, but I think that is the question that has been discussed by many groups who are working on risk assessment for these compounds.
Okay, cool. And then we have a question from Sergio, and they want to know, can ECOTOX be used to build a species sensitivity distribution? Will that SSD be the one that the chemistry dashboard is in the process of releasing? How can a representative endpoint be assured so that double dipping is avoided?
All right, those are also some really good questions. And data from ECOTOX can definitely be used to build a species sensitivity distribution. We don't currently have the option to build an SSD within the ECOTOX user interface on the web, but the export of the data is very simple, and we are exploring the option to have the data formatted, an export that is formatted for inclusion into the SSD toolbox that was developed by Matt Ederson. I'm not sure if that's the one that is being referenced here as for release on the chemistry dashboard, but that was recently released, and the crosswalk will be there. We have been speaking with Matt Ederson about the possibility of making an easier transition, although there are study quality decisions that are typically made before you decide which of the studies you want to include in your SSD, and that is something that is a researcher by researcher or a program by program decision that needs to be made. We feel that the information that are available in the ECOTOX database can help make those decisions, but we are not making them for you.
Okay, cool. I'm not seeing anyone typing, but I guess we can wait another couple seconds or so. If anyone has any questions, please feel free to just put those up in the chat box. And if you want to follow up I will be posting these slides online on our website later. I'm going to put the link to where those will go there in the chat. So slides and a recording of this webinar will be online at that site later today.
And I just want to do another shout out to Colleen Elonen, the ECOTOX coordinator. Thank you for letting me present all of your information today, and also thank you for being on the line. Don't know if you want to add any brief closing comments.
No, I wanted to thank you for doing an awesome job presenting my slides for me. We wanted to make sure we didn't have any technical difficulties by tag teaming like we usually do. But again, if any of you have questions for me or for Jennifer, you can send them to me. I do get the ecotox.support@epa.gov mailbox. That goes to me. I also have outreach staff, researchers, other biologists, people that can answer the questions for you. We have a team of about 15 people that work on the ECOTOX knowledge base, so there's a wealth of information there in history and people who know and can help you find the data if Jennifer and I cannot.
Okay, great. Well, I'm not seeing anyone typing or anything right now, so I think it's probably safe to say we can just end this a little bit early, give everyone a little bit of time back. But thank you both, Jennifer and Colleen, for presenting and sharing this information with us. Everyone, please be sure to turn in next month for a presentation on AOP.
Thank you so much for having us today.
Thanks for coordinating this for us, Sammy.
No problem. Hope everyone has a great day.
Yep. Bye-bye.
All right, bye.